Hello, everyone. Okay, let me see if we have anyone. I'm just going to try to grab the agenda. Oh, okay, found it. All right, I think we'll likely have a lot of items on the agenda today, and we're likely going to, I think we'll focus on the very first 4 presentations. We're still waiting for some people, so we'll just give them like 2-3 minutes to join, and then we can begin. Yeah, I think we're waiting for Taiwo. I think one more minute and then we can begin with the speaker that is on the call. Okay, so yeah, thanks everyone for coming to the October general meeting of the Africa BP. We have quite a number of items on the agenda today, so I hope we can finish them in an hour, or otherwise we can just stay slightly over today. We have some guest speakers for. Well, I just got a message from Taiwo who is kind of having internet challenges, so we'll just go to the next speaker. Do we have Nicolas on the call yet from PacBio? So we don't have Nicolas. Then we go to Ayorinde. Is Ayorinde on the call today? James.
Oh,
Yes.
hello. Yeah.
Okay, go ahead, please. Ayorinde will be telling us about the Gene Lab Bioscience and the Oxford Nanopore technologies. Over to you, Ayorinde. You can actually share your screen.
Okay. Thank you. Um, just a moment. Share screen. Okay. Good afternoon, everyone. Can you see my screen?
Yes, please. It needs to be in slide mode because it looks like it's showing the speaker section.
Oh, okay.
Yeah, so I'm kind of seeing your speaker notes. Well, not the note, but the back end of side of things.
Oh, okay. Okay. Let me stop
It's
sharing
just
it.
swap. Yeah, swap display. I think I think if you do swap display, that would solve it.
Okay. What about now? Okay. What about now?
This is better.
Okay. Yeah. So good afternoon, good evening, and good morning, everyone. Thank you for having me on the call. My name is James, the official leads for the Gene Lab Bioscience, and we are the official channel partners for Oxford Nanopore Technologies in West Africa. I have a very interesting topic here, which is latest genome sequencing and molecular biology techniques. And I'll just quickly jump into it. Then we can move ahead. Next slide. Okay. As someone, a scientist who has transitioned into the industry, I like to look at things from the perspectives of the economy. What are the African challenges and need for latest sequencing techniques? Of course, we have issues with climate change and drought, which is, causing different migratory issues and leading to the emergence or reemergence of pathogens. We have rise in cases of non-communicable diseases, NCDs. Most times when we talk about sequencing in Africa, we are talking about infectious diseases many times, but now we are seeing the need to also transition fully to genomic sequencing for humans to support, non-communicable diseases that are becoming, problems to us. There is need for us to, have information of local species for conservation purposes. Many times there are loss of some specific or local species or animals in our society that we need to have some information about them and improvement of agricultural yield. And all of these are probably challenges that are in line with, Africa's economic outlook for 2025. We talked about making Africa's capital work better for Africa's development. So if we can tackle all of these challenges, we can have significant economic improvement in the region. So going to the scientific topic of the day, right? I'm a bit biased towards some techniques, and I have them listed here. Interestingly, several sequencing companies have done quite a lot, to develop, sequencing technologies and applications that are useful, and most of them have evolved today. But the ones that keep fascinating me are adaptive sampling, whereby sometimes you just want to sequence some specific regions of the genome quickly and you can get data. We are used to targeted enrichment, where we'll spend time with PCR amplification or using Cas9 technology to cut specific regions on genomes for sequencing. One is real methylome classification. There are lovely papers out there now using epigenomic data to classify some kind of diseases. A good example is acute leukemias, and even for some CNS-related diseases. Telomere to telomere, for developing, full structure of genomic information for large genomes, especially humans, and long reads, from different long read vendors have helped significantly to help push this narrative and help resolve solving the genomic architecture of large genomes, which is also similar to Pore-C for lots of very large genomes, which someone from Oxford Nanopore would also be talking about. And lastly, single cell stroke spatial transcriptomics, which is one of the future, that is appearing now in the world of, trying to look at heterogeneity within a particular tissue, heterogeneity of cells. But what I'll be focusing on today, because having to talk about all of these different fascinating applications will take almost the whole of today. But what fascinates me the most, which I'm biased towards today, is the adaptive sampling technique, which talks about our ability to sequence what we want real time, without having to prepare PCR mix for targeted enrichment, hybridization pull down applications and the likes. And we can just move ahead to what adaptive sampling is talking about. And thankfully, we are partners with Oxford Nanopore Technologies, and it's a technique that has revolutionized or democratized sequencing. using the nanopore technology. And by what I mean by nanopore technology is that you have a beta barrel protein, etched on a chip, and you can have current flowing. And whatever flows through that pore based on the probes that are within the blue structure, which is the barrel protein, you can generate squiggle signals, which can then be converted, based on neural network algorithms to different, base calling or base calling formats, such as your BAM files, unaligned BAM files, or your FASTQ files. So why this technology? This technology has a very small footprint, it can be moved anywhere, it's very scalable, and you can get rapid, insights immediately while you're sequencing. And you can also get anything that passes through the pore will be sequenced if there's been a training model for whatever biomolecule that is passing through the pore. And what do I mean by this is that we are able to, get methylation sequence data as long as we are getting the, canonical basis real time while sequencing, which is a very good omics application technology. And the beautiful thing about it, again, is that it has, we're doing a long read sequencing, which helps you cover, areas where we might have problems that short reads might not be able to cover, your homopolymer regions, which help you resolve them, your haplotype facing, which helps you properly resolve them, and and ensures that sequencing can be done anywhere. And so I'll just quickly look at the devices just to, because when I started with the economic importance of sequencing in the region. And of course, that tells us that we need devices that are scalable, that we can take anywhere, and that are very affordable, that can be used anywhere within the region. So we have, different flow cells, and the mean ion flow cells with certain amount of pores, and the promethean flow cells with definitely more pores, and for sequencing output. So as small as the mean ion flow cells are, theoretically, you can get about 48 gig of data out of it, and your promethean flow cells can give close to about 290 gig of data. Um, um, so in adaptive sampling, people, I know a lot of persons here are very familiar with sequencing technology, so I used to, different approaches. But in this approach, you're doing your target enrichment electronically. So you're telling the Mino software that sequence what I need, and please do not sequence what I don't need. So how does that work? You upload your reference file, your FASTA reference file, and a bed file that indicates the region where you want to sequence. And once sequencing occurs, once there is, docking onto the pore and sequencing starts, and you have a region that is first sequenced quickly, if it doesn't meet the region of interest, then the strand is ejected. And if it meets the region of interest, then the strand can continue sequencing. So this is just to show, what the reference FASTA file looks like many times, and we have to be careful to ensure that the information on the header line, is similar to the information on your target bed, such as if you're writing chromosome 1, if you're writing it like CHR1 on your target bed, it should also be CHR1 so that it's easy for the system to look for the information, rather than in some cases where you have on your FASTA file where you have chromosome written in full and your target bed chromosome written in short form. So this is what happens in adaptive sampling. So we have the FASTA file or the reference file, which is represented by the blue color. And we have our region of interest, which we have indicated by the bed file, represented by the orange color. And we can see the sequencing strands falling in between our region of interest. But of course, if we look at this sequencing strand, or sequence strand rather, and we can see the adaptive sampling chalk outside the region of interest, but part of it is within the region of interest. We may have issues like, losing some of this information due to our adaptive sampling chunk not between the region of what we want. So there is a trick that if we can buffer, the sides of our region of interest so that our adaptive sampling chunk can fit in between this region, then we can have better on-target reads and coverage for our region of interest. So sometimes we may have off-target reads, but most times the off-target reads are ejected, but many times you see that we have enriched significantly the on-target reads such that we can quickly, do whatever analysis we want to do for the target region. Why do I like this approach? Take, for instance, I'm working on a metabolic pathway and I want to look at all the genes involved in this metabolic pathway. I want to quickly do a scan. So I can quickly design a bed file for all of these genes, in all my genes of interest, and I'll upload my reference FASTA file. And in no time, I can quickly generate an incredible tons of information, and I can do my analysis on time. Unlike before, that I would have to begin to design primers, for each of those regions, or be designing, hybridization experiments for each of those regions to pull down, which takes a lot of time for me to design validation of most of the experiments. But in this case, just my bed file, I get everything done. So it's a very simple library prep protocol. You don't need specific or special kits. The same ligation sequencing kits that you use are the same kits. You can also use your rapid barcoding kit as well. So you don't have to be looking for a special kit or, to run your experiment. It's definitely PCR free because once it's PCR free, then you can have access to native DNA sequencing,
Yeah.
which will retain. Hello?
James, just you have 2 more minutes, please.
Okay, thank
Yes.
you very much. Which will retain the methylation information. And well, it's in terms of cost, it's cheap, easy to update. You can keep updating your target bed file. And one beautiful thing about it is that someone will say that, oh my God, this is supposed to talk about the genome. You can have background genome information that you can use to analyze or provide copy number variations as well in addition to your region of interest. So I think due to time, I just want to say thank you for your time, even though I have some more slides to share, but I think I've shared the basic principles of this method. Thank you very much, and thank you for listening.
Yeah, thank you, James. So I think we'll also hear from Ashan on the Pore-C aspect. So I think for us in this community, our interest is more on the telomere to telomere and all the other things. So I don't know, does anyone have any question, comment for James? So James is the partner. I'm not quite sure how Oxford Nanopore says it. Is it channel partner or partner? But he represents Oxford Nanopore in West Africa or Nigeria, James?
Yeah, so
Um,
it's channel partner.
Channel partner, yeah, in West Africa. So, and they do ONT. So it's been a long time because we've been trying to also, understand, the ONT supply chain and logistics. But and I'm also pleased to see that it's taking shape now with Raksha. Raksha was with us 4 years ago, um, basically. So does anyone have any comment for James? Question, contribution? Otherwise, we would move to the next speaker, which we're going to move to Taiwo because he's now on the call. I'm just kind of trying to move this very fast because of the large number of speakers we have today, and I hope we can get to the other items. Otherwise, we'll leave it to next month. Any comment for James? Contribution? Yeah, so James, I think one comment from me essentially is of interest to us is also the policy you mentioned. So I know we've had this discussion over email. I mean, can you tell us a bit more, just in 30 seconds, if this is a procedure you've done before, or this is a procedure that you guys are willing to do, because we often have this ONT short read, long read method. And I think I can see Craig on the call. He uses ONT for some of our assemblies. And so do you just want to talk more about it, and perhaps Craig can just chip in just in 1 minute,
Oh,
both discussion, yeah.
well, thank you very much. So for the Pore-C, we are looking at genome assembly, where you do some form of digestion, and but it's a technology on ONT that it's relatively new, and we have one of the experts coming on the call this afternoon to talk more about it. But the advantage of long read sequencing protocol or devices is that helps you to feel that region. Now, if you're looking at the reference genomes, you now have access to T to T. I'm sure of that for the human genome, and that has been majorly due to the long reads. But in addition to the long reads as well that you can also get the methylome information, right away, because if you have the native DNA passing through the pore. So what I can tell you is that you can get your long read and you get your methylation data, epigenomic data right away. So for the Pore-C, the analysis is a bit different because the fragmentation stage is a bit slightly technical with some enzymes, and I don't have authority over that technology at the moment, but we are planning to bring the experts in country to run the first sample that we will be talking about tomorrow. I guess we have a meeting with you tomorrow about a sample on a genome assembly. And that might be the first one that I'll be doing in the region. But for now, we've not run any in the region. What we are currently looking at in the region is whole human genome projects within the region, which is going on well. And we are also doing adaptive sampling for now. Yeah.
Yeah. Thank you, James. So Craig, let me just bring in Craig here because he's one of the Africa BP coordinators and also doing heavy sequencing with ONT. So Craig, with the Pore-C, is this something that you guys have attempted in Cape Town?
No, we
Attempted?
haven't
Yeah.
attempted it as
Okay.
yet.
Okay, all right.
But I was interested in in the adaptive sampling because we've we've been doing adaptive sampling for reduced representation methylation sequencing for some of our human samples, but we've had now quite a few requests to do this in animal samples for animals as well. And I know for humans there's a standard bed file that one uses for the adaptive sampling. Do you guys at ONT have any plans to have bed files, custom bed files for other species?
Thank you for this beautiful question. So you can actually create your bed files by
Mm-hmm.
yourself, right?
Yeah.
And
So
you
we
have
were thinking of doing that and having sequencing the whole genome on ONT and then picking out the methylation sites. Like you said earlier, you get the methylation data. But if there are any um, plans for any of those already um, generated bed files, it will be awesome if you have any.
so if you also check on the community, I
Mhm.
think there are some persons that have run some adaptive sampling experiment, but not validated by ONT.
Okay.
The one that I know that is well validated is the hereditary cancer panel
Mm-hmm.
and the RRMS protocol for which is validated. But what
Mm-hmm.
we are doing in Lagos and some parts of Nigeria now is to work with customers to develop their own specific bed
Okay.
file
Mm-hmm.
based on interest. There is a beautiful, I don't know what your bias is, if it's for animals or human genomes,
Both.
um,
Yeah.
both, right?
Mm-hmm.
Yeah, but there was this beautiful paper for those doing leukemia studies, and they have lovely bed files published by I think a very lovely work, recently done, I think in North Carolina. I could share the paper with
Yes.
thank God. Yeah.
Mm-hmm.
So they have their own bed files. So you can look at bed files, that other persons have worked on. You can edit it. You can add your
Mm-hmm.
own bed files. And there is also a bed file checker to tell
Ah,
you if your
okay.
bed file was properly prepared. So I would share all of the links with thank God, and with that you could just try. So before you go ahead with the experiment,
Mm-hmm.
use the bed file checker. Be sure that your bed file is well prepared. And it's some people just use I think some CSV files just to prepare it, and or you can download it from UCSC Genome. If you can use the table of UCSC Genome to download the bed file of your interest.
Yeah,
So
so
it's something you can do by yourself.
so
Yeah.
I think we can
Um,
take this conversation,
mhm.
via email so that we can go to the next speaker. Thank you, James and Craig. So it looks
Very
like an
much.
interesting
Thanks.
conversation already. So I think I'm going to share once he sends the links to me via email. I would distribute it to the WhatsApp platform. I think this is more internal. Thank you, James. So we're going to move very quickly to Taiwo. Just for 10 minutes. Taiwo will just, I think after Taiwo, Ashan will then come back to you. Is Taiwo on the call yet? Taiwo?
Hi, thank
Yeah.
God.
Okay. Hi, Taiwo. Okay. All right. Taiwo will talk to us about an aspect of Africa, an interest of Africa BP, which is like the core fulcrum around permits and its experience, applying for permits, material transfer agreements to national focal points. So oftentimes uh, so just a bit of a background is that we have biological organisms in the environment. Human is also a biological entity, and for most human sample, I think those in the human space can correct me on this, is that you don't need you need ethical approvals, but you don't actually do need it's not like the biodiversity space. So here's the deal, right? In biodiversity, the biodiversity or the biological resource is community owned. So, and when it leaves the country, it's kind of a national thing, just like any other resources the country has. So that is how it's kind of classified, and there are several regulations that guide it. And unlike human, I think you can actually take your blood sample and give someone and they do experiment. It's more like for you, not community per se. So Taiwo will share his experience on getting that permit and the approval process and how he has done this. He has 2 permits to his belt already. And I've invited him to share his experience. And from next year, we'll move more deeper into this discussion, especially within the Africa BioGenome project. We've been having it before, but now we're kind of advancing it to go much more deeper. Over to you, Taiwo, please, 10 minutes, and then we can have 5 minutes Q&A.
Um, Good afternoon, everyone. I don't know if you are able to see my slides.
Yeah, you can see your slide, but you need to put it in slide mode, please. Slide sharing or something.
Okay, is this okay?
Yes, this is fine. Go ahead, please.
All right. So thanks a lot. Thank God for this invitation to share my experience, which is essentially on an experience on export, permit application approval, and the access and benefit sharing landscape in Nigeria. And this is about my experience so far. So I would focus my talk around this, where I'll have a disclaimer, and then ABS Nagoya Protocol, ABS Nigeria Framework, ABS Export Control Matters, and then the others as I've shown on these slides. So on the disclaimer, I want to specifically say that I am not a representative of the Federal Ministry of Environment. So this presentation is an account of personal experience from the application of export permit with the Federal Ministry of Environment. And that the presentation exclusively applies to situations where genetic resources leave national and continental borders. And just a recent discussion with with Thank God, and who had shared with me that there was a communication between the Federal Ministry of Environment, that's the ABS focal contact, and the African BP management, which reveals that permits may not be needed, or the MTA, which is Material Transfer Agreement, may not be needed where research is 100% carried out in the country. Are you with me, everyone?
Yes, please carry on.
Yes.
I can hear you.
Now, access and benefit sharing and the Nagoya Protocol, you all are familiar with this matter, especially that the Nagoya Protocol is an international ABS instrument that provides the international legal context for ABS, and that this is implemented at national levels through domestic regulations and competent authorities, and many more other issues that is available online with respect to access and benefits and the Nagoya Protocol. But here, talking about the access and benefits sharing and the Nigeria framework, it is benefit framework is implemented through the National Environmental Regulation, which is enshrined in the Federal Republic of Nigeria official gazetted document, and as displayed a section of it for you. This document is available online, and you could always access it anytime individuals want. And that the Federal Ministry of Environment, especially the Department of Forestry in the Federal Ministry of Environment, has been very instrumental in handling matters that has to do with the access and benefit sharing, especially with respect to export permits and for the country. The National Focal Contact Person for Access and Benefit Sharing and Export Permit application is, as displayed here, Ahmed Labara, who is the Chief Scientific Officer responsible for the process that one has to go through if needs to obtain export permit or a material transfer agreement for their biological samples. Okay, so the Nagoya Protocol ensures fair and equitable sharing of benefit arising from their utilization to the Convention on Biological Diversity. And this is usually documented where you have parties involved, countries, maybe collaborators, and the terms and conditions are always put in paper so that each party will be familiar with what is expected of them, and that what at that given time the government wants to be occurring. But the question then is, why is access and benefit important, especially the export permit and the restrictions that are there for each country? And it's because there's the need to protect biodiversity and indigenous knowledge, as well as national sovereignty over genetic resources, and also to manage biosafety and biosecurity risk and meet international obligations. And of course, important to encourage local resource capacity and reduce export of biological resources, especially the African BP is very much into this and ensuring that all things are done within the country in this respect. But just to give you a quick background of the project that led to the request for export permits in the case of my experience. So in 2011, I became a PhD student at the University of Oslo, and where I was expected to collect some samples for my thesis. And then I started, by nature of the project I was to do, had to collect sperm and blood samples from different locations in Nigeria. At the same time, collaborate with some other partners in Cameroon, to collect quite a number of samples which I'm to use for this project. So essentially, I was looking at the evolution of sperm competition and sperm evolution in African birds, but the focus is in Nigeria, where I had to sample species of birds from Nigeria and then also from other countries. And at the same time, I was interested in the DNA barcoding of birds in West Africa. And of course, extending this beyond boundaries, looking at sub-Saharan Africa. So eventually, all of these require that I obtain permit for this. But initially, this was not before me. So, and that's where I just briefly talking about the export permit application and what's the motivation actually. In 2015, when I was about to publish one of the papers for my study group, it was the reviewer and editor of the Biological Journal of the Linnean Society that first raised ethical concern on the paper and that was submitted for that group. What were the issues? So he pointed out that he is not aware, because they were not put in the paper, any permit we had for ringing or trapping the birds. That's one. And then he also pointed out that there was no indication that we obtained sample export license. And of course, during the period I was collecting samples, coming to Nigeria from Oslo, going back, and then going with samples, I never had any export permit. The only thing I had with the sample were just a note from APLORI, who would always introduce me as a student who was working, who was studying at the University of Oslo, and that I needed these samples for my project. But then at the airport, I had custom officers as well who would look at the samples and they would only ask me, what's my status? And then I get myself at that corner where I have to make explanations that this is what I am. I'm a student here, and that these samples are from birds, and I want to use them for my project, and I'll be using them in the laboratory in Oslo. And after this talk, they would always pass me and allow me to go with the samples. And that's how I've done for 3 consecutive years that I have harvested samples from Nigeria. And of course, this is also the situation for many other scientists who have always had that opportunity, collect samples, and then they can leave the country with their countries with these samples without much checks on them. So that then led us to begin to think of we need a retroactive export permit for samples that we already had collected, and at the same time get a new permit for the new samples that have not been exported to Oslo. So the first permit I had was for the samples already taken to Oslo, which was the retroactive permit that was obtained in 2015. And then this new one that I had in 2021 was the one I then had to use to lift the samples I had with me then to Oslo. Then what were the processes that I had to go through in the process of requesting the approval for export permits? One was I made contact with the focal persons at the Department of Forestry, Federal Ministry of Environment, and to discuss how I could go about this. And first point of the call was for me to apply and submit an application letter indicating my interest that I needed an export permit for samples. And alongside this, I was given a form, the ABS form. And the ABS form have its questions in there, which I need to fill in details. It was more like extracting the original proposal for the project and then putting bits and pieces based on the questions that they asked in that ABS form. So having filled those forms, I had to also include a complete list of species that I intend to, export the samples to the lab there. And then also the request of the Memorandum of Understanding with the University of Oslo, because I was from APLORI, AP Ontological Research Institute then, and then before I got the admission to the University of Oslo. So they were asking for Memorandum of Understanding between APLORI and then the University of Oslo, so it could be part of the processing. And then details of the stakeholders involved, the partners, collaborators, universities, and then for individuals involved, the CVs and means of identification of those individuals. In this case, the international passport was required. And my application was submitted 14th of November, 2020. And then that was it. So after the submission, I expected to hear back from the ministry, and then they got back to me around December, but then they would always want to request for additional information or seek clarifications where things put in the application was not clear. So having received that feedback from them on 1st of December, then it was for me just to clarify things and then formalize the payment of the fees for the permit, which I did following the year in 2021 and the 10th of February, thereabout. So I paid the sum of this 100,698, and that was just the fee for the submission of the application and every other thing. After that, they received it, they acknowledged, and then it took a few weeks. They invited me for an interview. That's essentially meeting with the members and staff of the Department of Forestry. And why they invited me was for them to interview me so they know what I want to do with respect to the proposal for that project, and then also state the terms and conditions for granting the permits. So on the 4th of February, yes, I was there. That was before the payment of the fee. So there with the Department of Forestry and staff, the interview was conducted, and then they stated their conditions for me, which is mainly to train the staff. And then they will also visit the site for inspection. And then they want to look at the samples that are collected. If possible, they want me to do some demonstration in the lab of what I will be doing. And following all of those will be the intensive training of the staff that were delegated for my own case. So that was it at the conclusion from that meeting and interview. And thereafter, I scheduled the training of the staff. I'll give you a quick one when I pass the slide. And then after this process of training the staff, there will be the check and clarification from the Federal Ministry of Environment. It's just to provide what they request and nothing more. And this is just for them to make sure that they are on the same page with you and with the application you have made and with your project and every other thing. And as well as if you have met the requirement, fulfilling and training all the delegates, and the feedback the delegate also provide to ministry to work with in developing the export permit. After that, you have approval, and then you will be invited to come and sign the material transfer agreement, which is also including the export permit that you requested for. And this will be done right there with the permanent secretary and the legal practitioners within the ministry. And then you have your witness who is with you. and you all go together to do all of this signing. Once this is done, you'll be given a copy of the NTA, and which is the permit as well. And you go through to then use that and report back to the Federal Ministry of Environment. That's why reporting is coming. And this can include maybe publications, a third-party transfer. If the samples you have exported, someone else indicated interest and want to use that, then you need to officially inform the Federal Ministry of Environment, because they claim ownership of these samples. And if there are cases of intellectual property rights, you also need to inform the ministry, and any other product that has arisen from the utilization of the samples you exported must always have to be communicated back to the Federal Ministry of Environment, as well as every other thing that you intend to do. So just to give a quick aside of what I've done, this is how the application I made was, but it's more comprehensive than this, but just to give you visualization of it, and which is always directed to the Permanent Secretary, the Federal Ministry of Environment, and then your attention, the Federal Director of Forestry of the ministry. And so thereafter, the ministry will give you back and invite you for an interview, as I discussed earlier, and this is always the letter they send to you. And in the meeting of interview and meeting with the department, this agenda is this is what the agenda will look like. Give you the opening, the self-introduction, the short brief by the acting director, then presentation by you, who is the applicant, and telling them about your proposed project, and then the further clarification on some of the gray areas that the staff, the Department of Forestry wants you to also talk more about. And most of the time, they'll ask you the benefit on conservation biology and on economic development of the country, adverse effect that the research may pass to the environment, or the modalities put together in place for benefit sharing arising from the utilization of the genetic resources as, as part of what they want to be built in the project. So that's just a quick one. So after that meeting, members, we had a group photograph with the staff, as you have here. And then this is just a receipt of the payment, the fees for the export permit. And they are very specific on the account you should pay this to, and they'll give you all of these details. Make sure you pay it directly to that account. If you pay to another account, you might end up paying another one again. And then for the training, I had this arranged for me to handle the training effectively. After the interview, we decided on the date of the training, then discussion with patrons. Then I made contact with resource person who helped with the training, and then procurement of materials, advert and publicity, invitation letters to participants, then reservation, accommodation, arrival of participant, training workshop head, then participant departure, all of this. So the budget for this were excluded from the fees paid. And then I made my schedules for the 3 days of the training, and this is what I covered with the resource persons I invited to also help in the training. So for that. And then after the training, these were just those on ground. We had a shot together, and members of the delegate from the Federal Ministry of Environment as well in this photo. And thereafter, they went back with the report, and they developed the NTA, and which is what they got back to me after a few weeks. And this was on the 10th of May, 2021, to ask me to request me to represent the University of Oslo, and where I need to officially still make contact with them to give me that authorization and letter so I could stand in for them. And after this, they invited me to sign the NTA, and here we are. I got this on the 18th day of April, which is what is written there, but it was already signed on that day. So here is how I went through to get the export permit for me. And I believe that not many people are aware of this process, and this serves as an eye-opener for those who want to export samples out of the country so that they could have proper export permits that could protect them as well as help ease the process of taking the samples out of the country to where they want to use it in the lab. And many other issues that I think were involved that I have not put together in this presentation that I will be available to discuss with people who will want to ask one or two questions. On this note, I'd like to thank you for listening, and thank you. Thank God.
Thank you, Taiwo.
Yeah.
Yeah, that's a good one. Does anyone have any question for Taiwo, please? Question, comment, contributions? Yeah, so anyone have any question? Anybody needs clarification? So it looks like everything is well understood on the process, right? Yeah, so we will be doing more of this. From next year, we're going to be launching the African Congress on Digital Sequence Information, and to kind of build more awareness and sensitization across the continent. And, yeah, but for us, one way, based on what we've just seen in terms of on the Africa BP communication with the national focal point, if you sequence the genome locally 100% and you don't ship samples outside, then no export permit, no MTA is required. And in a recent publication that Africa BP published for South African species that we published in July, South Africa has similar thing, but in their own case, you have to get the permission from the provincial government. Then, apart from that, you don't need any other thing. You don't need to have bioprospecting permit and all of those things. So from May, we'll begin to go, step by step to each country and begin to unravel this as well and kind of build more awareness. Thank you so much, Taiwo. We have just about 7 minutes or so. I'm sure we're going to run slightly over. Today, we are going to be listening to Ashan from Oxford Nanopore. Ashan will talk to us very briefly about the Pore-C in Oxford Nanopore. Ashan, over to you, please.
Sure. And thanks a lot. Thank God for the opportunity. I just want to confirm, can you see my screen?
Yes, we can see it, but we're seeing, can you just swap displays? Yes.
Yeah, there we go. Perfect. So yes, 7 minutes. So let me go through this really fast. But
yeah,
again,
it's okay if you go 3 minutes over. That's fine. If
sure.
just make it over 10 minutes. Yeah.
Sounds good. Sounds good. Thank you so much. Again, it's really a pleasure to be on this call. So thank you, everyone. I think James going before me is going to help me a lot. And thank you, James. I think he beautifully put together what is nanopore sequencing and the intricacies of adaptive sampling. And then Craig, you had some really interesting questions, and please do look at the chat. I did send some links that might be helpful. So okay, so what are we going to do in the next 10 minutes? As I said, my name is Ashan and I am a field scientist for Oxford Nanopore. I'm based in Dubai. And what I do is, kind of support the scientists across this region, build genomes. So I'm going to quickly break this down into 3 sectors or 3 parts. I'll explain to you what is Pore-C, and it's really exciting that you are going to do an experiment, and I think it's discussed tomorrow. Good luck with that, and hopefully this talk will kind of touch upon some points for you to think about. Then we're going to show you very quickly how do we set up a Pore-C experiment, some of the challenges as well that I want to make sure you are aware of, and then why do we need Pore-C and what advantage does it bring to the table. So again, thanks to James, I don't need to go through this, but the whole point here is that with nanopore sequencing, you're not limited by, the length of a fragment. So the limitation is actually the extraction. So the longer DNA molecule that you can extract, you can sequence on our platform. And what that enables is it really makes it easy to assemble genomes. So if you think of a puzzle, the bigger the pieces, the easier to put the puzzle together. So it's the same concept in genomics where the longer the reads, it's easier to assemble that genome. And that's what we really make use of when it comes to nanopore sequencing. And as James mentioned, we do native sequencing. So there's no bias, there's no PCR within this workflow. And what that means is you capture the epigenetic markers. So you don't need to do multiple experiments to get your genomic sequence, your epigenetic data. You just need to do one experiment, right? Now, in terms of the platform, how it's used, you can use it in different ways. And here we are going to focus more on the assembly aspects, right? So now assembly from our perspective can be seen as three different experiments. And Pore-C is one workflow within that, let's say, overarching experiment. So I'm going to focus more on Pore-C, but I do want to give you or touch upon what are the other experiments. So when you do a nanopore assembly, you have ultra long data from ultra long sequencing. Then we do something called polishing, which is the experiment B, and then you have Pore-C. Now, let me give you very briefly what each of them mean. So first of all, ultra long sequencing. I mentioned already, you don't have a limitation in length. So here, the advantage you're trying to take is longer fragments, easier to scaffold and create those contigs. So what I've shown here on the left-hand side is basically an example of an ultra long DNA sequenced. Here's the fragment profile. So your N50s are about 90 plus KB, and then you have long fragments up to even 600 KB. Remember, I'm not talking about base pairs, I'm talking about kilo base pairs. So these are really, really long fragments. And that just makes it easier when it comes to contigs and scaffold building. The second experiment is polishing. Now, what is assembly polishing? It's when you have homopolymers. So there are parts of the genome where you have a whole lot of A's or a whole lot of T's, right? One after the other. Now, it's always difficult for sequencing methods in order to tell exactly how many of them exist based on the techniques. Now, what we do here is we introduced a modified A or a T. So what that means is, since in nature, in a human genome, you don't have A's and T's that are modified. So when we collect that squiggle signal, when we collect the electrical data, we are able to, identify those modified bases. And by doing so, we are able to get a good read of those homopolymers. So that's the polishing bit. Now, the third experiment is Pore-C. So Pore-C, if you really want to simplify it, it's almost like looking at the genome in a three-dimensional perspective. So what is chromatin conformation capture? It's basically looking at the genome, as I said, in 3 dimensions. But what you're trying to identify is parts of the chromosome that are close to each other and parts of the chromosome that could be way further apart. And also in a 3 dimensional space, interchromosomal contacts. So let's say chromosome 1 is close to chromosome 3 in a 3 dimensional space, right? So that's what we get in Pore-C. So how do we set up the experiment? I am going to give you an example from human blood, but again, this can be done for all sorts of different species. Now, just briefly, the protocol itself, what you do is you need live viable cells. You do a form of cross-linking. So you have your histone proteins that are close to each other, stuck to each other. Then you permeabilize the cell so that you can send through reagents inside the cell. So remember, at the beginning, you're doing something called an in situ digestion. So that's keeping those, you're digesting the DNA fragments, but while they are fixed. And then you have proximity ligation, where you're ligating them back. But here, based on proximity, based on distance between molecules, you are ligating, different. So you're creating a polymer or a concatemer of different parts of the genome, right? So this is not just a single read. Now, in terms of the protocol, again, one thing I already mentioned, having viable cells is key. So you need to make sure these are viable. It's a 3-day protocol. I'm not going to go into details. I'm happy to share these slides with anyone who requested. And what I have highlighted in yellow just means you need to think about that step. From my experience of doing Pore-C, you spend more time actually preparing buffers. So make sure you have all your buffers ready. Make sure you have your viable cells. But if I was going to give you a tip, it's all about planning the experiment, because it is 3 days of work. So the first day you cross-link your histone proteins, then you do a restriction digestion. Next day, you do the proximity ligation. After proximity ligation, now you're digesting, whatever the histones and so remember, you have some 18-hour incubation steps, which is why it's a 3-day workflow. And then in the last day, now you have created these concatenamers that you will sequence. One thing, please remember that you are using formaldehyde, phenol, you are using things like chloroform. So make sure that you have all the right setup when it comes to safety. At the end of the day, we want to say, keep ourselves safe, right? In terms of resources, Nanopore community has plenty of them. So please feel free to access them. It will guide you through the different paths. Now, just finally, very quickly, why? Why do we do all this? Why do we do Pore-C? It's all about creating contact maps. And there are, in an assembly perspective, there are three ways of looking at it. Number one, it will improve your scaffolding, right? So what you're creating is a multi-way contact. Now, how does it improve? Now, just imagine you have a very long DNA strand, and now you have a variant in one side. Thousands of base pairs later, you have another variant. Now, if they're in close proximity in 3-dimensional space, you can be sure that this is in a single chromosome, and that helps when it comes to haplotype phasing, right? You're able to tell the maternal and paternal and phasing it by using these long range data. Number two, especially with the repetitive regions that are present in genomes, these multi-way interactions can really help you resolve them. And lastly, errors. There can be errors, misassemblies when you assemble genomes, and especially when you're looking at interchromosomal contacts. So by creating these contact maps in a 3-dimensional phase, all you do is long range phasing and correcting for misassemblies, and all that is the benefit of doing a Pore-C experiment. Hopefully that was a quick rundown of Pore-C. Just to let you know, there's a lot of talks online available on YouTube, by the way. Again, there was a talk from Dr. Temitayo looking at cattle, from Dr. Lee looking at plants, and then from A-Star Singapore looking at human genomes. So comparing, hybrid approaches to nanopore approaches and giving you the flavor. So in summary, I hope I give you a very quick rundown of what is Pore-C, how do we do it, and why do we need it. Hopefully that helps.
Yeah, thank you so much, Ashan, and for actually kind of breezing this through. Does anyone have any question? We have one final speaker from PacBio. Does anyone have any question, comment? So Ashan, I'm sure we will dive more into this deeper tomorrow with James. I'm not sure if you'll be at the call or something. Yeah. Anyone have any question? Craig, you are very heavy on ONT. So do you have any question, comment?
No, I don't have any questions, but I would like, when Anthea is back, maybe to put her in touch with the guys from ONT, particularly. We have some cross-linked samples that we want to sequence, and we were thinking of doing it on IC in MGI, but if we can use Pore-C to do those, I think Anthea will be very happy, because I can already see those chromosome level assemblies would be a lot easier than the one that we did for helichrysum. But I'll put Anthea in contact with you, because I see Ray's hands up and I don't want to.
Yeah. Okay. Okay. I think Ansia can just Ashan. Craig, I will just put you in CC after the meeting with Ashan, so that when you write when Ansia is back, you can just reply, so that you have Ashan email as well. Okay. So Dr. Ashan, thank you so much for your presentation. Please, there's a question or questions in the chat. Please answer them, because we may not have enough time, so that we can move to. So Ray, one question very quickly. 10 seconds. Over to you, Ray.
Okay, no, thanks for the talk. I just wanted to ask, how does the Pore-C solution in terms of OmniC, how does it support the phasing? So would it help with reducing the need for trio samples in that respect? Thanks.
Yeah, definitely. And I think also remember, this is a multi-way contact. So what you're going to get from a polymer that we create out of all these different contact points is going to help with the phasing, and that will kind of reduce the needs for trio in samples. Now we also see that not just with Pore-C but also ultra long sequencing, experiment A, where we see in situations where in reality sometimes you don't have access to parental DNA. This kind of helps when it comes to removing that need of trio. So ultra long sequencing can also become a solution for that.
Yeah, it looks like there's so much interest in the process. So I think we can take this conversation via email. I think Ray also, if you need, I'm sure Ashan and James will be at some point. We'll invite them over for one of our workshops and give more detail. From what I can tell, there's a high interest. So we can move to Nicolas. Hi, Nicolas. Yeah.
And if we are over time already, we can move it to next month. I don't mind at all. It's up
So
to you.
we have next month is full. I think we have 2 other providers. So if people happy to stay on for 10 minutes? Yeah, okay, let's do that, Nicolas, so that, because next month we already have other scheduled speakers, so it might be a bit challenging. Let's do this, please.
Let's have this. Okay, now.
Yes,
Okay.
yes.
Give me 1 second to present. Just a second.
So I think we have three speakers from some organization speaking here.
Do you see my screen?
Yes, please carry on.
Okay. So I wanted to talk to you shortly about what we can offer at PacBio and what will be the new stuff that will come out during the first half of next year. And because we have no time, I will be very quick about it. So what we can do with the Revio system at the moment is that you can start from, with the Spark chemistry, you can start from 500 nanogram of input DNA and get between 100 to 120 gigabytes of data with a base quality of 90% at Q30 and more. And you get, at the same time, you get 5MC and 6MA data directly of this. And you can use this technology to do something that is quite similar to what was described before, which we call sci-fi. It's a new workflow for a single library chromosome scale assemblies. So it's based on the HiC protocol, where you can detect close DNA interactions directly. And what you do is you do exactly like the HiC protocol, but instead of getting single interaction between 2 positions, because you can create long multi-contact fragments, which are still cross-linked for some of them. You just amplify them with our new amplify protocol. And from this, you get a hi-fi library with different segments that can help you to get full, to improve read mapping and haplotype phasing, especially in repetitive element. And because of the amplified part of the project, you can use a sub-microgram low input to get chromosome scale assembly of single organism. And I have an example here that is a preprint that was published this year, where you can see one sci-fi read at the bottom, and this sci-fi read contains different part of the chromosome 2 that you can see, and it aligns around the complete chromosome so that you can follow very far distances across the chromosome, and it allows you to assemble a full chromosome very quickly. And just as was described before, what you can do is, because you can phase your sample, you get haplotype phased chromosome for each of the chromosomes. So in this example, here we see all the chromosome phased, and we can even detect a chromosomal rearrangement between some of them. This workflow, as it's described in this paper, require 2 separate libraries, but we have a new workflow where you can just prepare the amplified fragments together, and because they are barcoded automatically during the amplification process, you can add them directly to your whole genome sequencing library. And with one SMRT Cell and one SMRT Bell library prep, you get all the information. And I'm here today because our Inkaba, our partner in South Africa, will soon start proposing to give this as a service. So you can ask them, and they can do this workflow for you. So that's the first part of what I wanted to present about this sci-fi technology. And also I wanted to present you about what's coming on the PacBio innovation roadmap, and especially the new chemistry coming for the review in 2026, which we call the Spark Next chemistry, which will add a few new things. So the first thing that it will give you is it will allow you to get 5-hydroxymethyl-cytosine detection automatically in every run. In the sequencing data, you will have this automatically. But what I would like to concentrate on today is we will be able to give you access to much lower cost hi-fi genomes by allowing you to use SMRT cells more than once to reduce sequencing costs dramatically. And so for a human genome, we think that this will allow you to get down to $300 to $350 per genome by sequencing 2 genomes per SMRT Cell per run, and reusing the SMRT Cell. So it's like 30 to 40% less than today. This will arrive probably in Q2 2026. How does it look like? So you put your SMRT Cell on the instrument, and automatically at the end of the run, the SMRT Cell will be washed so that you can start on the next run. You can start with the same SMRT Cell, with the same library or with a different library. It doesn't matter, without having to do anything special with the SMRT Cell. The instrument will do everything automatically. And to show you the kind of data that you can obtain after one use or on the second use on this graphic. So you can see, for example, on the top left, the hi-fi yield. We get between 120 we get more than 140 gigabytes of data with this human 15 KB library. on the first use, and we see only a very small decrease in total yield for the second use, going around 120 gigabase of data. This is across 28 different smart cells that were sequenced. In terms of quality of the reads, we are still well above 90% at Q30 or more bases. So this doesn't change between use 1 and use 2. The read length in KB remains the same, very similar, around 16 KB for both libraries before and after the use. The 5-MC methylation, of course, remains the same. And if we look at, for example, variant calling in human at 20x coverage, we get exactly the same. So we get 99.9 F1 values for single nucleotide variants. We get 98% for indels and 97% for structural variants. And also CpG island with allele-specific methylation, you can see the numbers are the same between use 1 and use 2. So we are very excited about this new release that will come then, and it will be also available at INCABA. So if you want to, starting mid next year, you can start to get this reduced cost for sequencing. And actually, I was very fast because I didn't want to take any of your time. So do you have any question on any of these?
Yes, I have a question. I will try for us to finish in 3 minutes. So it's actually interesting to see all this development coming up. So one question that I had is, I don't know if I misheard it or something, is that does the process allow you to, because one of the challenges has been doing hi-fi, doing hi-C or whatever, and then having 2 different steps. Does that process allow you to do everything in one go? I think I misheard it. I just want to confirm.
So you still need to do so you have your genomic DNA. You still need to separate it into 2 tubes at the beginning to do to get your cells to do the DNA crosslink for the hi-C part of the protocol. But once you finish this part of the protocol and you've amplified your DNA, you can reunite it with a whole genome sequencing library or with genomic DNA, and then do only one library prep and sequence everything together. So usually what you could do is to do 90% genomic DNA, 10% hi-C, and you put everything together, you do one library and you sequence on the rest.
So which means you have like everything can be sequenced in one run and then you separate it.
Yeah, exactly.
Okay, that's a good one. Does anyone have any question for Nicolas? That was quite interesting. Any question? Ray, I think, yeah, I know you would have because you guys are doing this. All right, over to you, Ray.
Yeah, no, thanks for that. Currently we're using Hi-Fi and OmniC workflows for our assemblies. So I think it would just as a comment, I think that would be helpful to combine it into a single process, because I think on our side we sometimes have to wait over long periods of time before we get our confirmation chromatin data. So yeah, that is a useful advancement. So thank you.
Yeah. So hi, Jonas.
Yeah, Jonas, you want to share? Yeah. So that's it. Yeah, I didn't want to present this part because I was not too familiar with it. So yeah.
Jonas, okay, very quick. Yeah, go ahead, please. It's nice to see you again, Jonas. It's been a while. We can't hear you if you're talking.
We don't hear you, yeah.
If you're speaking, we can't hear you. You're mute.
Now we see you.
Well, we can't hear him. We can't hear him. Yeah, so I think let's why we're trying to hear him. Does anyone have any question? I think this is going to be interesting. So Nicolas, if privacy allows, do you want to share your slide with me? I mean, you don't have to.
Yes, I will, of course. No, I will. Yeah, no worries. Yeah.
Yeah, okay, all right, thank you. So that we can share with the community as the...
Yeah, exactly.
Okay, Jonas says, don't know why, just showing the picture of what Nicolas was talking
Yeah,
about.
exactly.
Okay,
So
all
that's
right.
what I was
Okay.
talking about. You
Yes,
do
yes,
the shearing and your high molecular weight was here, and then you have the 3C protocol on the other side. You
yes.
amplify the DNA, and then you do one library.
One library. Actually, this is good. This is good. It's going to reduce the runtime as well, or the time it takes. Okay. Let me not keep people waiting. Thanks. Does anyone have any question for Nicolas or Ashan or Taiwo? Any final question, comment, contribution before we end for today and then reconvene next month? Any comment? Otherwise, thank you so much, Nicolas and Ashan and James and Taiwo for the presentation. And I'm sure it looks like there's quite a huge number of interest in the Pore-C. And as we reduce the time, as the time goes from 3 days or wherever to doing it once, I think that will also help particularly. And I was going to ask you one question about those that have Hi-Fi, how that integrates into it, but I don't want to take people's time. I can do that via email much later. All right, see you all in... Jonas, do you want to say something? It looks like you're trying to say something. Okay, all right. Okay, see you all in November. All right, bye-bye.